can live a long time with carcinoid cancer, but will eventually succumb to the disease. Its 5-year survival is approximately 67%. And there's a lot of people that are actually walking around that don't know they have carcinoid cancer. So it's been predicted that the walking diagnosed patients is closer to 100,000. Interesting, there's no approved drugs for this cancer. People are actually treated with supportive care to mainly alleviate symptoms that are called carcinoid syndrome, which is basically flushing and diarrhea and things like that, with saminostatin analogs. There is a phase 3 trial that's ongoing by Novartis, actually. It's a triple drug combination, and that's in phase 3 right now. So this type of cancer, based upon the data I showed you earlier in terms of some of the in vitro work, it's predicted that this is going to be less sensitive excuse me, to small cell lung cancer based on both the in vitro and in vivo testing of carcinoid cell lines. Nevertheless, we should hope to see some response. And we did. So if you look at here, so actually here's a large tumor mass that, and this patient actually received, I believe this was 10 to the 9th, and 6 weeks after treatment, this tumor mass, as well as this one right here, is completely gone. And so the bottom line is we actually do see responses like this. And another thing that we can actually do when we do have the opportunity is we can biopsy the tumor after treatment. We can actually show that virus is there, and you can see active virus in the tumor. So we saw 3 near PRs by CT, stable disease with decreases in chromogranin A. That usually goes up or stays high when people have cancer, and so it would go down if there's actually a response. No responses, identified progressive disease once. We had an approximately 70% response rate. And again, as I mentioned, it's important that all these patients actually had long-term stable disease, and as I mentioned, all the patients going into the trial actually had progressing cancer. So very promising data, and that was actually not at the high dose. And when we actually go to the high dose, the data actually got slightly better. I want to just point out one patient in particular, patient 23. Again, no dose-limiting toxicity, and this was at the highest dose tested. And the post-treatment PET scan showed a 50% reduction, a PR according to the GIST-PES criteria. And not only that, she discontinued her pain medication, and so she was definitely symptomatically improved. Her chromogranin A results also decreased, and she has stable disease. At this point, she has stable disease. So we also have some more pending data, and so 10 out of 12 of the patients in this cohort had clinical benefit, either stable disease or partial response. And this is just that PET scan I was telling you about. So if you look, here's a couple months later, and so this is actually the amount of activity actually in the tumor in terms of FDG glucose uptake, and this is actually what it looks like 2 months later. So in summary, for the carcinoid, it actually buys them weeks, if that. So it's actually a very serious disease. And again, as I mentioned, the types of patients that were entered into our trial had really failed both first and second line therapy, and they had overall survival rates of weeks, not months to a year. However, small cell lung cancer is actually predicted to be extremely sensitive to this particular virus based upon all the in vitro and vivo testing we've done, and also looking at primary cultures. As I mentioned, the patients in the study had failed multiple lines of therapy and had a very low quality of life. And because of the safety issues with this group of population, we essentially only treated patients at the low dose of 10 to the 7th. Nevertheless, the promising data from this is that 1 out of 5 of these patients who had progressive disease going into the trial and had failed 3 lines of therapy actually still alive today, more than 2 years after they have gone on this treatment. And if you look at the data overall, we see a 3-fold increase in overall survival. And it's difficult to compare this because there's very little data on third-line therapy in terms of looking at life expectancy. But if you do look at second-line historical data, and so for instance, this is actually the benefit that topotecan gives you in terms of progression-free survival or overall survival here, if you look at these two lines. And so you can see here, there's a difference here about several weeks, and here progression-free survival is about a month. And this is our data, and it's actually out farther here, but you can see that we had a substantial progression-free survival tail with prolonged clinical benefit and no toxicity. And I should mention, topotecan is an extremely toxic chemotherapeutic, as well as cyclophosphamide, the first-line, etoposide, the first-line therapy. Many people actually die from the chemotherapeutics, approximately 5% of the people. And this patient actually, as I mentioned, is out 24 months. The patient is still alive here. It's one out of 5. So from the end of this trial, we talked to a lot of key opinion leaders, and we decided to run a phase 2b consolidation study in small cell lung cancer. So basically, this would take patients that have just received frontline therapy with etoposide platinum, and we'll treat directly after that treatment. And that way they'll have low volume disease. We'll treat at the highest dose, 10 to 11 becquerels per kilogram. Our primary endpoint will be progression-free survival, as well as overall survival as our secondary endpoint. There'll be 100 patients in the trial. 50 will get treated, 50 will not. We'll be treated with placebo. And we'll be looking for overall survival in this group, and we would expect to actually hopefully see a therapeutic benefit, and we'll also do things like monitor biomarkers such as integrin alpha-4. So this trial has actually been approved by the FDA, and North Central Cooperative Oncology Group will be running this trial soon. It's expected to start in approximately 1 to 2 months. There will also be a trial that will be run by the NCI. That trial has also been approved and should be starting shortly. That's using this virus to actually treat kids actually with cancer such as neuroblastoma, retinoblastoma, medulloblastoma, things like that. And again, that will actually be a phase 1 study, again doing some dose escalation, only at this time we'll be starting at a dose of 10 to the 9th becquerels per kilogram. That's kind of where we are in terms of clinical development. And in summary, we believe we have an impressive safety database from all the species showing no dose-limiting toxicities. And again, the primary adverse event was mild flu-like symptoms lasting usually for 1 to 2 days. We did see replication of virus in small cell lung cancer, longer-term survival, increase in overall survival and progression-free survival. And our key opinion leaders are highly supportive of utilizing NTX-010 following first-line therapy in small cell lung cancer and doing a pivotal placebo-controlled comparator study. In carcinoids, we saw PRs, near PRs, and clinical status improvement observed in multiple patients, and as well as multiple doses, 10 to 11 looking at like it's the best dose, and progression-free survival in the low-dose groups compared to best phase 2 studies that are out there performing other drug combinations. And again, there's a lot of people that are also interested in doing work in carcinoid cancer with this indication. However, it's very difficult for a small biotech company to actually do something in this indication because you'd have to be like a 5-6 year study to look at overall survival when you have a 5-year survival rate like in carcinoid cancer. And with that, I'd like to thank essentially the people at Neuroendocrine involved, as well as Charlie Rudin, who was the PI of this phase 1 study at Johns Hopkins, as well as Harvard, Baylor, several people at the NCI, as well as US Oncology. Thank you.